let's think about more natural systems. Everything that we've looked at up till now has been a highly constrained, a very artificial laboratory system designed so that we can follow what's happening analytically in a very clear and simple way. But of course, the vesicles that form naturally aren't so nicely constrained. They're extremely heterogeneous, and that generates a system which is much harder to study experimentally. So that's illustrated by this image, which shows the range and the diversity of vesicles that are formed when you just shake up oleic acid with some salt and buffer. You get these huge vesicles, tiny vesicles, compound vesicles. It's a terrible mixture. And if you imagine adding some more oleic acid to this in the form of micelles so that everything grew a little bit, you couldn't tell that anything had happened. So we need new analytical ways of looking at this, or new ways of preparing vesicles so that we can actually watch what's happening. And our major advance in that area came from the work of a new graduate student, Ting Zhu, who had a background in mechanical engineering and proposed a very simple and elegant way of preparing more or less uniform populations of vesicles that were large enough to just look at in the microscope. So for the first time, we were able to do experiments where we take vesicles, add food, and just watch how they grow. And so what you can see in this image is a field of vesicles. They're all roughly 4 microns in diameter, so big enough to image optically. And what you're actually seeing here is a fluorescent dye encapsulated in the aqueous internal space of each vesicle. So now what you'll see next is a time-lapse video of how these things actually grow following the addition of more fatty acids. What we expected was simply that these spherical vesicles would swell up a little bit. We just watched them get bigger. We thought that if the surface area grew faster than the volume, they might become somewhat elongated in shape. But we did not expect what actually happened, and which is quite dramatic. And so what you see here is initially a faint filament emerging from the parental spherical vesicle. And over time, that filament grows and absorbs all of the material in the starting spherical vesicle, which has now grown into a long, thin, branched, filamentous morphology. So this was a very surprising observation. It immediately raised a host of mechanistic questions, which we're still investigating. But it, very importantly, immediately solved the problem of how cell division might happen in a manner driven by environmental fluctuations. As I said, spherical vesicles are hard to divide. It takes a lot of energy. But these structures are incredibly fragile, and all you have to do is shake them up. Gentle agitation is sufficient to cause them to divide, and you can see that process in action in the next video. So here you see the purling instability. The filament forms a set of beads, and then they snap apart and generate fragments which eventually round up into spherical daughter vesicles. And so this process is driven by very gentle perturbations in the fluid, very mild shear forces. You can easily imagine the shear forces generated by wave action on a shallow pond leading to cell division in this kind of process. Now, this process of growth followed by shear-induced division makes a cycle that can be iterated indefinitely. So you can start off with a spherical vesicle, grow it into a filament, divide it into spherical daughter vesicles, which can grow again. If they sit around, the volume increases, they round up, then you can grow again, divide, grow. this cycle of growth and division can be carried on indefinitely.